Hi, hi everybody, and I'm very honored to introduce Art Weis to the seminar. Art was supposed to spend his last sabbatical visit here in Montpellier in our team at ISEM, starting in January, but he had to delay his arrival because of the pandemic situation. So hopefully he will arrive here mid-February, maybe, if indeed we are out of this epidemic wave. And so it will be around until April, end of April. Is that correct, Art?
Yes.
Approximately. Yes. So do not hesitate to reach out if you want to meet Art in person when he's around. And I'm very honored too, because I've been reading about his work for a very long time and got very impressed and interested by everything he did. So that's really interesting for us to welcome here. So Art is a professor at the University of Toronto. He told us that he's about to retire in a year or two. It has a long career behind him, and is worked on a lot of topics about, I'd say broadly, plant biology and reproduction and evolution. And among his recent work, and I think things he's going to tell us about today, is worked a lot about the evolution of phenology of flowering in plants, and connected to issues of adaptation to climate change, and more recently also assisted gene flow. And he's also among the first that have used the technique of resurrection ecology to study evolution in the context of contemporary global changes and plants, with very interesting and successful results. So maybe he's going to tell us more about that as well today. So he's been doing theory, experiment, a lot of different things. And so we're really happy to hear him today and excited. Thank you, Art.
Okay, well, I will start. And thank you very much for that kind introduction. I'm very, very disappointed that I can't be delivering this seminar in person, but there's lots of things we're not doing in person nowadays, and I've become somewhat resigned to doing many, many things by Zoom. And let's hope that this goes off without a hitch. Okay, so what I'm going to talk about today is a series of experiments that people in my lab have been doing since I arrived here at Toronto about 15 years ago. And I just sort of bring together a number of empirical studies, and they're all built around trying to figure out causes of natural selection on plant phenology. So if you think about how natural selection in natural populations has been studied over the last 30, 40, 50 years, what you find is that there's a whole lot of documentation that selection occurs, a little bit, well, a lot less about verifying the causes of selection. And the documentation of selection, I mean, its founding goes back with this particular method anyway, with George Price of the Price equation. And this is a piece of the Price equation right here, which describes the strength of selection, covariance between fitness and some trait. If you use breeding values here, then you are measuring the response to selection. If you put in phenotypic values here, you're measuring the strength of selection. And what this says is here's what the statistical relationship is between fitness and the trait. And if it's a positive covariance, then you can say that there's directional selection for increase in the traits, negative for decrease in the trait. The ambiguity here, though, is the variation in Z the cause for the variation in fitness, or is it some other trait which happens to be correlated to fitness, correlated to Z that's causing this variation in fitness? Russ Landy and Steve Arnold sort of fleshed this out a little bit, again, saying that the strength of selection on trait Z can be partitioned into the direct selection, that is the causal link between the trait value and fitness, and the indirect selection, that is any selection through causal values exerted by other traits, say trait Y, using the partial covariances and the covariance. Okay, and this was a very, very important advance. I'm sure many of you listening to this have actually implemented this sort of equation in this format somehow. The limitation is, though, is that this is a very strong method, gives you a very clear picture of what's being selected on if you have all the traits. And when do you know you have all the traits? Well, you don't know you have all the traits. And so as Landy and Arnold and others of the Chicago school emphasize, is that, what this really does is describe a statistical relationship. If you really want to know the causes of selection, why does an increase in trait Z lead to an increase in fitness? You really have to do some sort of manipulative experiment. And so this is something that I've been doing on various points over the years with people in my lab. And so I want to tell you about some of them. And the trait that we've been interested in most is flowering phenology. And there's lots of possible ways in which flowering phenology can be acted upon by selection. First, there is, of course, whether or not the plant is able to attract pollinators. When a plant reaches a new habitat, you might have to find an adjustment in flowering time to suit the new conditions. As the climate changes around you, it of course affects whether or not flowering on one date is better than flowering on another date. And then there's all these other things, including how phenology can be involved in reproductive isolation and how it mediates gene flow. Okay, so flowering time's an important trait to work on if you want to look for an example of selection and dissect out its causal elements. So basically, this is a little diagram I have that's sort of sets up the situation here. I work on annual plants because they're easy. And the point is that what we want to know is, when is the best time to make this transition for an annual plant between vegetative growth to actually turning on the flowering? And if you think about it here, that there's a T sub 0 is the time of germination. T sub F is the beginning of the flowering period, when the plant is actually putting out pollen, receiving pollen. And then after pollination, then there's time for seed maturation. So there's all these elements here: the time spent growing, the time spent reproducing. And it's pointed out that there could be a trade-off here, is that the longer that you spend growing, the bigger you are. But then what that means then is that you have less time to actually complete the reproductive process. And so that's one of the things that we're going to be talking about first. But there's also the external environment to think of. And when you're talking about what could be the selective pressures going on to select on flowering time. And if you look across the season, of course, in the temperate zone, you have a peak of temperature in the summer. You have rainfall that might be sporadically dispersed throughout the year. Pollinators, the peak abundance of pollinators might appear at one time. The peak abundance of natural enemies might occur at the other time. And so if you have a plant that starts to grow at this point and flowers on this date, this is what we would call the general environment that it's exposed to. And you can see it's slightly cooler, but it's got pollinators and it escapes the enemies. If you flower at this time, you're not getting the enemies, but you're also not getting, the pollinators, but you're also at a maybe more favorable temperature. So there's this general environment that is going to determine the fitness. And then you have also these internal factors, this trade-off in time between time spent growing versus time spent reproducing. And so this plant, by flowering early, has lower capacity to produce seeds. This one flowering later might have a higher capacity to produce seeds. So that's sort of an internal trade-off that will have a causal effect on fitness. And then you also have the idea here that when you flower is going to determine like who you're competing with. Okay, so if your flower is sort of too early, you might be limited by the availability of mates. Too late, again, mate limitations. If you flower like with everybody else, sort of in the middle of the season, you might have lots of potential mates, but you'll also have more potential competitors. And so this sort of social environment then can also influence selection on when you do things over the course of the year. So I like to think about plant flowering phenology as being similar to habitat choice. Okay, it's not just that flowering on a particular date or at a particular age is good. It's by flowering at a particular age, you flower at a particular time of year that happens to be a good time to take advantage of a good environment. Okay, so going back again, general environment is what you interact with, social environment is who you interact with, and the internal environment, which is about the acquired resources and how they're distributed out, that's about how much you bring to the match. So I'm going to talk about two things. I'm going to talk about trade-offs in the internal environment and how that trades off the time of flowering versus time spent flowering, vegetational seasonal shifts in the general environment, and then we'll talk about competition. And instead of talking about flowering time, then we're going to talk about germination for part 3. So let's go to this first one. And again, here's the idea about this trade-off is that we have this plant flowering early. It doesn't get very big before it flowers, but it has plenty of time to produce all its seeds, and it produces, seeds. This one flowers later, it grows bigger, but it runs out of time of maturation, and many of the seeds that it could potentially produce fail, and only some of them then actually mature. So you might imagine then there could be some sort of intermediate flowering time that would yield the greatest seed production. Indeed, we did a model. I'm not going to go through the model. I'll just show this equation to let you know that I did a model. And basically, what we found is that the optimal flowering time depends, of course, on how long the season goes. And this is a model that builds upon what other people in the past, like Rothgarden, has done similar sorts of things. And one of the things this shows is that if this is the distribution of flowering time, that the fitness function for flowering time varies according to how long the growing season is. 30 units, 28 units, 26 units. You notice that under a short growing season shifts the optimal flowering time to an earlier date, a longer growing season to a later date, an intermediate. The other thing that comes out in this is that you have an asymmetrical fitness function. And that's an interesting sort of thing, because what that means is that if this is the peak, the average flowering time right here, and the flowering time average lines up with the peak of the fitness function, you still nonetheless get selection for early flowering. And that's because flowering later than the peak it leads to lower fitness than earlier than the peak. And you can see that here. Here's for a long growing season. What that means is that if you flower this many days after the average, you have a modest increase in fitness. If you flower the same number of days before the average, you lose a lot of fitness. And the idea here is because of this asymmetry of the fitness function, that flowering too late is bad. Flowering too early is not so bad. So anyway, so the to test this sort of idea, we did an experiment with strains from Southern California, and we imposed 2 watering regimes that mimic the California growing seasons. A short growing season stopped at 51 days and a long growing season at 88. In Southern California, the growing season for brassica is determined by the rainy season. And sometimes the rainy season is short, sometimes it's long. And this is an experiment done by a number of people with me, including these people below. And basically what we saw is that if you have a short growing season, indeed, you get this sort of strong selection for earlier flowering. If you have a long growing season, you get, in this point, we even detected like an intermediate optimum, so weak selection for later flowering. And you can see how this corresponds to these 2 fitness functions. So we think then that this is pretty good evidence that the length of the growing season is going to be a big factor in the evolution of selection on flowering time, particularly if you have seasons getting shorter. So what does this have to do with the real world? Well, I'm going to talk about this experiment in resurrection ecology that was referred to. And this is an experiment that just depended on a couple of real lucky breaks. This was not at first intended. We were just lucky with this. It starts with Denise, who was a graduate student studying plant defenses against herbivores and pathogens. She went out to collect seeds. I told her, collect some seeds for your experiment. I sort of envisioned that she would collect that many seeds. What she collected was that many seeds. And so we had all these seeds left over after she was all done with her dissertation. She quite wisely then put them into a cold, dry incubator, and they sort of sat there for 7 or 8 years. 2004, I heard a radio report, and that was the California water officials are becoming alarmed over our 5th consecutive year of drought. In a Mediterranean climate, drought, what means is that the rain starts more or less in December. The rainy season, instead of ending in April, the rainy season ends at the beginning of March. So you have a shortening of the growing season under drought in California. So this gave me the idea then that we could do something like this. So here are the 5 years preceding the drought. We have the seeds that were collected by Denise. Those are sitting in the freezer. Shenna went out back in 2004, collected seeds from the plants that are growing there, which would have been then the descendants of these 1997 plants. So we had these ancestors, we had these descendants, we can grow them together in a greenhouse side by side after putting them through a refresher generation, and asked, has this 7 and 4 or 5 generations of drought selected for earlier flowering? Grow them up in the greenhouse, and what we found, indeed, that's the case. So we had 2 populations. One was a population which the soil was always very dry, very sandy. Those plants flowered earlier anyway because of the dry soil means that it dries out earlier in the spring, inherently shorter growing season there. Growing season got a little bit shorter. You found a 1.9 day advance over the time. And in the Arboretum population, where you have richer soils that hold onto their water much later into the spring, you still had an advancement then in flowering time over this period of time. So going from ancestors to descendants, and we actually made ancestor-descendant hybrids, and the hybrids fell right intermediate between the ancestors and descendant parents. So it's pretty strong evidence that, yes, this idea that the length of the growing season is a cause of selection on flowering time, I think is pretty well established here. Okay, so short seasons favor early flowering despite the smaller size with early flowering. Now, that's looking at the internal environment. What about additional causes of selection on these plants? And the seasonal shifts in the availability of pollinators, in climate, and all those sorts of things, they can also exert pressure on flowering time. And so Emily Austin did a dissertation on this under my direction, and I'm going to talk about that right now. Okay, so the thing is that we wanted to know, is do these sorts of factors up here exert additional selection on flowering time? So what that sort of says is that it's not the age of flowering, it's the date of flowering that determines what sorts of things in this external environment that the plant encounters. The trouble in studying those is that it's inherently correlated with the age at flowering. If you flower at an early age, you also flower at an early date. So what's important? Is it the date that you flower or the age that you flower? And that's sort of a conundrum that we tried to solve. And if you look at it this way, here's what the internal environment would be important, is that you flower at this particular age. It's going to be, you're going to be able to produce a certain number of seeds. But if you flower at early, you're small. If you flower late, you're large. And so that's going to be a factor in the selection on age at flowering. And then the temperature, pollinators available, that's going to affect total seed production. And the point is, what's most important? Is it this direct effect on production of age, or is it the effect of age on production through these correlated factors here, through the size, or is it through the external environment that the plant has, quote unquote, chosen by flowering at that particular age? Measuring these sorts of things is very problematic, but this is the general environment, and general environment is going to have an effect on seed production. Genes that control age of flowering are going to have an effect on seed production because it exposes them to a particular set of factors in the general environment. We can't measure those things directly, but we can measure the date at which they flower, because we know those things go up and down over the course of the season. So the question we wanted to know is, how important, do you find any effect of age at flowering on seed production once you've accounted for the date? But as we said before, these two things are inherently correlated. So we want to try to break the correlation. And so we can manipulate that correlation between flowering age and flowering date simply by staggering the sowing date for the seeds in the experiment. So to give you an example right here, imagine that we have a genetic line that takes 45 days to flower, another one that takes 21 days to flower. Well, if you plant on the Julian date, 152, that's the 152nd day of the year, then you can expect this to flower at date 197. Similarly, with this line, You can get it to flower at date 197, but if you flower it later at date 176, so that you can get genetically young, genetically old lines flowering at the same time. This is a young plant coming into flower. This is an older plant coming into flower, but they're coming into flower at the same day. We had lines like this because we ran a large greenhouse population through a couple generations of very, very strong assortative mating. And we were able then to have some plants that were genetically young. We knew seeds from these mothers would flower quickly. We knew seeds from the other mother would flower late. And then you could end up with these sorts of correlations where you had age of flowering and date and flowering perfectly correlated, negatively correlated, and uncorrelated. And we could then plant these out into the field and determine whether or not the selection on age changed depending upon the sign of this correlation. So in other words, what we want to do, we made this correlation positive as it is naturally, negative, which is exact opposite of the natural situation, or zero. And what we want to see is, does this change predictably with this correlation over here? Does change in the genotype environment correlation change the selection gradient on age at flowering? So I'm going to show you a graph that gives the answer, but I need to explain what you're going to be looking at in that graph. Here we have plant age and relative fitness. So this would, the slope of this line would be then the selection gradient on age of flowering. If the slope is positive, what that means is that the older plants are favored. If the slope is negative, that means younger plants are favored, and the slope is zero. That means there's no selection on age. Okay, so what we want to know is whether or not you get these differences in slope as you change the correlation between age and date. So there's three possible outcomes that we envisioned. First is that direct selection on age of flowering is all that operates. In other words, it's good to be young when you flower, regardless of the date. Here, it doesn't matter what date you flower. Simply flowering at a young age is not important. It's the size that you are when you flower. So all the selection through age is through its effect on plant size at flowering. And then finally, you would expect something like this. If the age at flowering is important because it gets you to the right part of the seasonal environment, that you expect a correlation, something like this. If the correlation between age at flowering and date of flowering is positive, you might expect then negative selection, which would indicate flower early. Over here, because the young go last and the old go first, then you would get positive selection for later flowering. And here, if there's no correlation between the two, you'd expect no selection on age at flowering. It doesn't matter if you're young or old. It doesn't determine when you hit the general environment. Does not depend upon your age. Okay, and so you expect some sort of relationship like this. Okay, so the question is, what did we find? Well, we did it over 2 years. And in the first year, what we found is that when there's a positive correlation between the age and date and flowering, so this is the natural sort of situation, that indeed selection favored those individuals that flowered at an early age. When it was the negative correlation, selection favored those that flowered at an old age, and that's because old went first. And if there's no correlation, there's no selection at all. Got it again in the second year as well, a positive relationship like that. So why do you get this very strong relationship in the first year, not the second? Well, in the first year, we had these guys show up late in the flowering period, caterpillars and weevils. And so plants that flowered very early managed to escape these. Those that flowered late did not. We sprayed insecticide in the second year, and the effect got much reduced. But still, the point was, is you change that gene environment correlation, and you can flip the direction of selection on the age of flowering, which means then that in addition to these internal trade-offs, you also then have selection acting through this general environment, being an additional cause of selection on flowering time. Okay, so this is basically what we concluded here. This correlation is naturally positive. When you made this correlation positive experimentally, then what you found is that there's a negative selection gradient on flowering age, which means younger is better. And the fact that that changes when you change this correlation means younger is better simply because younger is earlier in the season. Earlier in the season is better conditions for the mating and maturation of seeds. Okay. To flower early is to choose the best environment in which to flower is the message off there. Okay, now want to turn, well, I'll just say right here, I talked basically about female reproduction here and the female component of fitness in these hermaphrodites. We worked also on the male component. The results there are a little hard to interpret. We're still not sure exactly what it all means, but at least through the female component of fitness, we're pretty sure. Okay, so turning now to the idea that, when you make a life history transition, depends upon the fitness consequences of making a life history transition at a particular time is going to depend upon how many of your conspecifics make the transition with you versus before you versus after you. In other words, the transition from one stage to the other depends somewhat on the social environment. And what we looked at here is an experiment where we could manipulate a trait, and the trait that we can manipulate in this time was the time of germination. And we know that time of germination is going to influence the intraspecific competitive environment for plants like brassica, which tend to grow in almost monocultures. If you look at this little pot here, here's a bunch of seedlings that have germinated. And what you'll notice is that this one germinated several days ago. It's growing up. You can see that it's beginning to cast shadows down here onto the soil surface. Compare that with this little plant here that has germinated just today. Okay, it's growing up in the shadow of its competitors. And so part of the advantage of germinating at a particular date is going to depend upon whether or not, how many have gone before you. Okay, if you are late germinating, you may be facing then a tough competitive situation. This is a form of what Bruce Wallace called soft selection. And Bruce said that when habitat quality sets a limit on a population's reproductive output, a mutant's fitness depends upon the density of the population and its rank in the local competitive hierarchy. Okay, so implementing this idea, illustrating it when it comes to germination time in plants, what you can think of is something like this. Imagine that we have time going this direction down here, and we have a series of plants that are all uniform in their germination date. And this day, everybody germinates, everybody's above ground. As they grow, they get bigger, they start competing more intensely for light and for soil resources. And finally, they come into maturation. Now, in this sort of situation where everybody germinates at the same time, you get a more symmetric form of competition between these. So these neighbors are suppressing the focal plant there in the middle, but then again, the focal plant is suppressing its neighbors as well. And you get a moderate variance in reproductive output because of this symmetric competition. If you have an asymmetry, though, so that one plant, a new mutant, then flowers or germinates early, what that means is that when the rest of the population germinates, it will already have a head start. That puts it higher in the competitive hierarchy, and it actually grows bigger. And in the end, it will end up suppressing its immediate neighbors. And this is an asymmetric competition. If you're thinking about just light competition, this plant in the middle is shading out its neighbors, but the neighbors are not capable of shading out the focal plant. Okay, so let's think about this in a broader sense, what this would mean about the intensity of competition and the variance in fitness and the selective advantage of a germinating early versus late. So let's imagine that this mutant, this early germinating mutant, is rare in the population. And what you're going to find is that some of the plants in its immediate neighborhood will be suppressed. Plants farther away will be experiencing symmetric competition. It's only these ones that are experiencing this asymmetric competition. You can even have a situation is because these ones are suppressed, those neighbors end up getting a little benefit by being a couple steps removed from this new mutant, but because there's only so few of them, very, very few of these plants get that compensatory ability to grow because of suppressed neighbors. What you would find in this case that the variance in fitness will be rather small. As that mutant spreads, though, what will happen is that you'll get a very, very large variance in fitness, and that is the early germinators will be surrounded most typically by a lot of late germinators, and that you have here the highest level of asymmetry and competition. But then as that mutation goes to fixation, suddenly you get in a situation in which like early germinators are competing against early germinators, and the symmetry of competition returns. Symmetric competition predominates, asymmetric competition predominates, symmetry returns. Okay, so we did that, and again, an experiment with brassica, and we set up some artificial populations, which varied the frequency of the early and late germinators allele. And what we did is we did a phenotypic manipulation. Again, we took plants seeds and randomly planted them on one day. Those were the early germinators. And the other plants seeds we planted a week later, those are the late germinators. So this was a phenotypic manipulation that mimics what a mutation would do if it accelerated germination by 7 days. Okay, and so when you do an experiment like this, you're manipulating the social environment by manipulating your germination planting date, but then you also have a general environment to deal with. Those that germinate 7 days earlier are going to have a different slice of the seasonal climate. They're going to have different pollinator availabilities. They're going to be exposed to different enemies. So there's going to be a general environment here, social environment here, and both of these can potentially exert selection on germination date. This would be selection, what would be called hard selection, soft selection here through the social environment, hard and soft. Okay, So again, this is an experiment done with a number of people, Kyle Turner, Emily Austin, Susanna Wagmar. And this is sort of a indication of how we set this up. We had these large they were actually children's swimming pools that were several centimeters deep, a meter across wide, filled them with potting soil and laid things out in this hexagonal grid. We had low density, we had early density. We had 100% early germinators, 100% late germinators, and then a 50-50 mix. 3 replicates of each round here. And we just wanted to see, what is the relative fitness then of what's the absolute fitness of early germinators versus late germinators when they're in these situations of low competition, high competition, over here, symmetric competition, in these two, asymmetric competition here in the middle. And basically, here's the fundamental results. Here we have number of pods that each plant's produced at low density. Here, what we found is that, if you are an early germinator and you're growing only with other early germinators, you had made this many pods. Late germinators growing only with other late germinators, you made slightly fewer pods. If you are in this mixture, what you found is that going early gives you a big advantage, and going late gives you a very, very strong disadvantage. You can see here then this asymmetry and competition. Now, in a similar pattern over here. Now, if you look at this and ask, well, what is the total reproductive output of these populations? You'd see something like this, so that it seems like a population of early germinators makes more seeds than a population of late germinators, and a mixture makes an intermediate number of seeds. What this would indicate is that, yes, the seasonal environment for the early germination was better, that there is some sort of hard selection going on for germination time here. And here you can see essentially the same results, just with everything all mixed together. Total seed pods made by these populations as a function of the frequency of the late germinators. If you're all late, you do worse than if you're all early. So there is a signature here there of hard selection. But then again, there's also the signature of soft selection as well, is that the relative advantage of being early versus late depends upon whether or not you're competing symmetrically with others like you, or asymmetrically against those that are different. And we can sort of quantify this difference in fitness between early and late by looking at the Genie coefficient for these populations. Now, the Genie coefficient is a statistic that comes from economics. It measures income inequality, and it sort of asks if every individual in the population is making the same number of seeds, and you went to the first plant and you say, how many seeds does this make? And then you go to the first 2 plants, how many seeds do they make in combination? If you go to the first 48 plants, how many seeds do they make in combination? If you go all the plants, how many seeds do all the plants make? You get this straight line if everybody made the same number. But if you do the same thing when there's variation, you get a curve that looks something like this. Here we have a situation where we had, this is one of these populations which had symmetric competition and low density. And what we find is that the Gini coefficient for inequality is low. But here we had high density and asymmetric competition. And what you find is the Gini coefficient is very, very large. As a matter of fact, over half the planets didn't produce any seed pods. And so you have this very, very strong measure then of reproductive inequality. That's a signature then of soft selection, where you stand what you do for fitness depends upon your rank within the population. And so the Genie coefficients for high density tended to be large, and they peak then in the intermediate frequency for the two types. Lower Genie coefficients for low density where competition was strong. So what this says over here on the two ends, those are the conditions under a symmetric competition. These are under asymmetric competitions. This then is a signature for soft selection. Okay, so how can you partition then? How much of the selection on germination time is due to the hard component? How much of the selection is due to the soft component? What's the relative importance of these two types of selection? I knew that I'd be close to the time here, so I didn't prepare all the slides to take you through all the algebra, which you'll probably be glad of anyway, but I'll give you a quick flavor of how you partition this out. And it's to use a methodology that's actually originated in sociology. It's called contextual analysis. And it's very much related to the multivariate selection gradient analysis of Landy and Arnold. So again, what we want to do is figure out how much of the selection is generated by the general environment. What were the physical and biotic conditions that the plant experienced when it germinated early versus the social environment? What about its competitive environment? How does that influence its fitness? So we know we have evidence for that. We know we have evidence for that. The question is, which one is more important? Is it the hard selection or the soft selection? Again, we can go through and use this covariance structure that Landy and Arnold originated, take an extension, borrow an extension of this, or a similar take on this that was developed in the social sciences. And what we would say is the covariance between fitness and phenotype, that is the number of seed pods produced versus the date at which the plant germinated, is going to be then due to the selection gradient on the day of germination times the variance. That's just a way to recover this covariance. So this would be the selection at the individual level. Plus, this is a piece that is provides the context for this covariance between individual fitness and individual phenotype. Here is the regression of seed production onto the average seed production in the population. So capital Z here, capital Z, that's the average value for Z in the individual population that they were experiencing. So the variance in Z times this regression coefficient then becomes the covariance. And these two together make up this total covariance between the two. So this is sort of like a correlated trait, if you will. Your context in which you're growing is a correlated trait to the trait that you're actually trying to determine the strength of selection on. And partial regression, individual fitness over individual phenotype, partial regression of individual fitness over population mean phenotype. Okay, so I hope that sinks in for what that means. Now, here's the thing. Charles Goodnight determined that under purely soft selection, these two coefficients, these two regression coefficients, should exactly cancel each other out. If selection is purely soft selection, they cancel each other out. So I saw that and said, well, that means then if these two added together don't equal 0, then there's something else operating besides soft selection, which must be hard selection. And so by doing some rearranging and thinking about what the components are, I was able then to come out with this partitioning of the total covariance here in between germination date and seed production, and come up to the conclusion that in this case, what we're finding is soft selection predominates, that it's about twice as strong as the hard selection component. So what this means is that in a species like brassica that grows up largely in almost monocultures, that you end up with who you're competing with being a very, very large component of selection on this trait of germination time. And if you're in a group that's very uniform in germination time, selection on germination time is weak. If you're heterogeneous for germination time, selection is strong. Okay, so just taking that together then is what we say is early germination had double the advantage of better growing conditions and superiority in asymmetric competition, but only with a scant effect on mean population, mean fitness. That means soft selection was strong, hard selection was weak. So, just a couple of simple take-home messages here. First take-home message is it was a lot of fun doing this stuff with these people. And what we found sort of indicates is that isolating the fitness effects of habitat choice from condition, that is the size at which you make this transition from vegetative to flowering, your body condition at the time, it was really very, very dependent upon genotype environment correlation. And so that what that means is that these sort of transitions over the course of the season from one life stage to the next are really sort of a form of habitat choice. The other sort of more general lesson that as an old guy giving to a young person would be that, if you want to understand the causes of selection, you got to look beyond doing just selection and gradients. You have to actually go out and do some experiments. And, me saying that as an old guy is the same thing as these guys were saying it as younger guys. They're old guys now too, but I knew them when they were younger. Is that, as important and as stimulating that the selection gradient analysis has been to the evolution of evolutionary ecology over these past several decades. If you really want to understand at an organismal level what causes selection, you have to take the next step further. You have to go do the experiments. Okay, and so then I just want to end up by, again, thanking all these wonderful people that have worked with me on these various sorts of projects. And also acknowledge financial support in the US from National Science Foundation, Canada from NSERC, University of Toronto, where I am now, University of California, Irvine, where I was until 15 years ago. Here's my email address. Here's my Twitter account. And I'll be happy now to take any sort of questions you might want to toss at me. Thank you.
Okay, thanks, Art, for this very stimulating talk. So we don't have questions yet in the Q&R tab, so people can raise their hands if they have questions. I would have quite a few questions, but some of them would be a bit technical or things that I didn't completely understand. So while we wait for other questions to show up, I'm sure Ophélie will have questions too, but I can sort of start with everything. I was wondering, because I've seen you use the Genie index, but I was wondering whether this index that come from more social science, things like this, does it capture something different than the variance does? Because I haven't seen the formula, I've seen the definition before. But.
Well, the alternative would be something like the coefficient of variation, would measure more or less the same thing. it was originally used by people in forestry when they were trying to plant out tree farms and figure out what's the spacing that would give you the best spacing and the most uniform types of trees in the most compact way. And it's I think it has a different sensitivity to skew and kurtosis in the distribution.
Something about monopolization then.
I'm sorry, what?
Something about monopolization or something like that.
Yeah. So if you have a platykurtic, platykurtosis, I'm sorry, leptokurtosis. If you have a very, very strong peak, you get a different Genie coefficient. You have a strong peak and broad tails, the Genie coefficient tends out to be more informative than just the coefficient of variation.
Okay, thanks. Ophélie?
Yes, thank you, Art, for this very nice talk. And I had a question about the last part. So in the treatment where you mix the different genotypes, they are evenly spread, especially. And I would imagine that due to limited dispersal, when you have a mutant, there's still some special clustering of genotypes in the population, and that might diminish this self-selection effect because you're competing more with your own type due to this limited dispersal. So I was wondering whether the conclusion that self-selection is actually much stronger than the other type of selection would be somehow softened by the fact that you've got this special structure in the real population.
Right. Yeah. And you're absolutely right. So these were randomly distributed and on a hexagonal grid. And so that's, let's put it this way. We set up the experiment where we were most likely to find an effect. and in that situation, the effect comes out strong and clear. In another situation, in another spatial arrangements, you may well find a weaker signal. Yes, that's absolutely true.
And actually, I had the second question, if there's no other done again.
Okay.
So you mentioned that you had looked at male fitness, and I was really interested into that because I think this is something often we don't really think clearly about. What is this selection on male phenology? Flowering phenology is different than selection on female component of fitness through flowering time. And I was interested in knowing what you found in the end.
Well, in those same experiments, Emily was able to evaluate selection on male fitness actually in 3 different ways. One of them was using microsatellites to determine who was the father of all the seeds that were produced in that experiment, and trying to figure out if early or late fathers were more likely to contribute. And what she found is that it didn't depend upon what date you flowered, that actually the selection on flowering time through male function was directly on the age of the plant at flowering. So young fathers sired more seeds than old fathers. And we thought that's rather odd. And, how could you end up that way? And we wondered, well, maybe there's a gene that influences siring success of young fathers that influences siring success because it gives the pollen grain a competitive advantage. So it doesn't matter when in the season you transfer your pollen. If you're young, you have highly competitive pollen. And so we develop experiments that doesn't work. So we're really kind of at a mystery at to why we get that result. And because we don't really understand it, I figured I would let somebody ask. And I was sure somebody would.
Okay, next we have a question by François Lefebvre. François, can you talk?
Can you hear me?
Yes.
I can hear you, François.
Okay, thank you very much. I was very much interested in your talk, and very nice. And in particular in the last one, I had maybe a silly question because, and maybe I mixed something. It's about the role of asymmetric competition, because actually asymmetric competition, somehow it increases the environmental variance it's a component of environmental variance actually. And then it somehow reduces the heritability for a given variance. So by the way, you show it has an impact that it increases the intensity of selection. But what at the end, if it really reduces the, let's say, the heritability, what is the final result in the response to selection?
In this particular case, the intensity of competition is correlated with the phenotype. In other words, you end up in that mixed situation in which who are the ones that are at the bottom of the competitive hierarchy? They are the ones in which have the simulated gene for flowering, for germinating late. Who's at the top of the hierarchy? It's the ones that have the simulated gene for flowering early. So in this case, you're not only amplifying the environmental variance in phenotype, but you're also amplifying the genetic.
The genetic, okay. Okay, it's not a random environmental, it's kind of genetically related environment. Yeah.
Right, because remember, early germination, has as its correlated trait is competitive superiority. And so those two things acting together, you can say competitive superiority is a pleiotropic effect of the gene for early flowering.
Okay, thank you.
Or early germination, yeah.
I was wondering regarding the second part of your talk, whether this impact that you find of the correlation between age and date of flowering, how this extends to perennial plants, and what would be the equivalent trait to the age of flowering. These types of correlations exist also, I think, for other types of phenological traits, for trees, etc.
There are other traits besides.
I think for longer lived plants where it's not the age that would matter, but perhaps other things in the.
Yeah, and so I guess it kind of depends upon what you count as age. And if you were thinking about a perennial plant that goes dormant during the
Exactly. Yeah, that was my question.
Yeah, so in that case, you could, instead of saying instead of the date at which say the leaves flush out, fully expand out, it would be what's the number of days between when the physiological switch to break dormancy is thrown. When you flip that switch, how long does it take then before leaves become fully flush? So again, that's a number of days. That's sort of like the amount of elapsed time between the initiation of the process and the process becoming complete. So that's sort of like age, whereas the date at which all this occurs is date. It's external to the plant.
Yes, so just going a little bit on this, I had the impression that for a short-lived plant or an annual plant, the trade-offs that you mentioned would be perhaps more extreme because they start from nothing and then they have to develop the whole plant. So have you been thinking a bit about this, how these trade-offs play out in different types of?
Yeah, and you know, there's been a fair amount of modeling done on these sorts of things, and the details are not right at my fingertips right now, but a lot of the models have looked at, you know, looking at how resources wax, or, you know, increase and decrease in availability over the season, and how do you set the timing to match those sorts of things. And you know, for some perennial plants, the switch to turn on flower development for next year occurs at the end of the previous summer. And so the flowering switch is thrown, say in August or September, but the flower doesn't actually open until May. And that's a way in which, you know, the resource for growing is available throughout the summer. So you grow, but the best time to actually produce the flower is a short span in the spring. And so you get that sort of, but you have others that start accumulating the flower later in the season. You have, if you have a different profile over the course of the season of resource acquisition and availability, then you can switch these things back and forth. And that includes both, you know, when you grow and when you get the resources to grow.
Okay, thanks.
Yeah,
that's interesting. We have a question by Nacho, I think. Nacho, did you raise your hand?
Yeah, I could. So I mean, I found this extremely interesting. I wanted to ask to know your opinion on whether there may be some directionality that imposes some ratchets to evolution. I mean, for instance, I'm establishing the analogy with the evolution of size in fish that are overfished, so that we are selecting for smaller individuals, right? With overfishing. And then what I have, so what is usually said is that this selection may be harder to revert than the opposite one, right? So that there some of the effects of selection may actually be imposing ratchets that are asymmetrical in the two.
Right.
And so I would like then to know your opinion on how because you were speaking about drought in California, on the reversibility on the short term and on the long term of these differences.
Right. This is complicated by the fact that not only is there this asymmetry in the fitness function that you would expect, turns out that there's also directional dominance in flowering time here. And so that if you select for the earlier, you get a much faster response than if you select for later. And that's because of the additive genetic variance in one way is easier to exploit than in the other way. But you know, I don't think there's been a lot of work on symmetry of fitness functions. I don't know a lot of work. One of the first instances I remember of it, though, is a selection on litter size in mammals. And again, there's where the mother has to care for so many mammals, and then, you know, little babies. And the idea is if you have one baby, they grow really big. If you have two babies, well, they both go pretty big. 3 babies, yeah, they all do okay. But if you have 10 babies, none of them survive, and you get then this asymmetrical fitness function for that as well. And so I think you're right, is that these asymmetrical fitness functions are going to cause things to go in one direction quicker than they will in the other. But then again, this also takes up or got to take into account whether or not there's directional dominance in the trait that you're looking at.
Okay, I think we, I just put a reference that reminded me of this discussion. So I think we're going to stop here because we're running out of time and not running out of questions. I think actually I wrote down lots of things, but the good thing is that luckily we'll have an opportunity to discuss this with you during your visit, hopefully. So I just want to, so thank you again.
Thank you. And
I
just hope to be sharing a glass of pastis with you all soon.
Oh yeah, with Ali. So I just want to let the audience know that next week we're coming back to the usual schedule of 11:30, and it will be Diego Fontanetto.
Okay.
So have a nice weekend, everyone.
Thank you very much, everybody.
Bye.